I'm Matt Drew, VP of Sales and Marketing at Rooster Bio. Over the next 10 minutes, I'll provide a brief Rooster Bio intro and give you a glimpse into why this company formed a short 8 years ago and what we have enabled to date. Before jumping in, I'd like to acknowledge the Alliance for Regenerative Medicine for inviting us to speak. The Rooster Bio team greatly appreciates the opportunity. Rooster Bio set out to fuel the rapid commercialization of scalable regenerative cures, and to do so by radically streamlining multiple stages of research, development, and clinical translation. So what exactly does that mean? To understand, we start by analyzing the typical timeline to progress from R&D to first-in-human testing. Over the course of this 7 to 11-year span, much time is spent validating raw materials and processes within a platform that enables you to test hypotheses. Your goal over this time, apart from finding clinical significance, are to have well-characterized raw materials and processes enabling you to achieve consistency and performance, to have an end product that can be manufactured reliably and at the required scale, and to put yourself in a position to achieve regulatory approval. This must all be done under constraints limited by time, money, and resources. Unfortunately, significant portions of these 7 to 11 years aren't spent solving the science. They're spent solving the supply chain, and in doing so, slowing down and reducing the ability for critical cures to come to market. Rooster Bio asked, what if we could address portions of this time, money, risk, and energy spent on supply chain considerations? Could we compress product development timelines and create a clear path from research to cGMP manufacturing? And what would a Rooster Bio fueled supply chain with off-the-shelf products and engineered processes look like? Most importantly, how would this all look in practice? Our hypothesis is that a supply chain with off-the-shelf solutions will ultimately make treatments cost less money to develop, take less time to progress to first-in-human testing, and that faster iterative progress in disease treatment will be made because major hurdles costing time and money will have been removed. And in effect, we can radically alter the landscape of regenerative medicine. Over the next few slides, we will review what is at the core of the Rooster Bio model and show how we have applied it to HMSCs and EVs. In the simplest terms, we do 6 primary things today. As experts, each takes us considerable time and energy. Therapeutic programs that attempt to do this on a program-by-program basis encounter considerable risk and opportunity for significant error, costing time. The first thing we do is acquire donor material under strict testing SOPs and regulatory guidelines. Rooster Bio manages the complexity around tissue sourcing, which takes significant time, effort, and diligence. Typically, programs at the R&D stage won't have the know-how and/or resources dedicated to consider tissue sourcing. Conversely, programs preparing for phase 1 trials are actively considering source material, but struggle with cGMP processing costs. This decision point, regardless of translational stage, impacts many aspects of a therapeutic program, but represents only a part of the challenge associated with generating bulk starting cellular material, which brings us to the second thing we do, which is generate highly characterized master cell banks and working cell banks. Acquiring the donor material doesn't guarantee a releasable master cell bank. Once cells are isolated and an MCB is generated, the resulting cells still need to meet lot release criteria. And a percentage of generated MCBs fail, meaning the already significant money and time associated with MCB generation can be sunk costs. The risks increase substantially when manufacturing cGMP MCBs, and the time and cost considerations continue to increase as programs progress through clinical trials. For example, at phase 3, qualification of multiple cGMP donors will be required, which we address by making multiple donors available as an off-the-shelf product. The third thing we do is productize those WCBs at a consistent population doubling level, with the right format and grade for each stage of therapeutic development. Having a consistent PDL is a key factor affecting how the cells perform and important towards making standardized processes. Each manufacturing lot of WCB is vialed or bagged into the proper concentration for the grade under which it was manufactured. 1 million and 10 million cell vials for RUO use and 20 million cell vials for cGMP use. There are also fully expanded, ready to print cells at the 50 million cell per vial level. Number 4 is we develop highly productive medias. A conventional bioprocess media with an analogous cGMP version would take you only so far into commercialization. Scalable medias need to be built with bioreactors in mind, which is why Rooster Bio media was built for bioreactors and then scaled backward into 2D. Medias focused on helping researchers achieve commercial scale also must provide maximum productivity while preserving cell health and function. The 5th step is combining cells and media into defined bioprocesses that scale consistently from 2D to 3D, maximize cell expansion and EV production, remove risk and complexity associated with status quo bioprocesses. And I'll stop here for a second because creating MCBs and WCBs has risk, but we've also made standard parts of the manufacturing process easier and less risky. For example, our media is fed batch, which means no media exchanges are required. This greatly reduces contamination risks and simplifies bioprocessing protocols. After removing complexity, we also reduce time and cost. And sticking with the media example, removal of media exchanges also means you spend less money on media and less time exchanging media. Imagine the time and cost and complexity required to change media as volumes scale up. Number 6 on our list is we support the regulatory approval pathway via Type 2 drug master files. Supporting trials with Type 2 DMFs means that the FDA has working knowledge of our products and that when you go to generate your CMC, large portions are already established. This holistic, unique approach towards manufacturing reagent supply allows therapeutic programs to leverage our bulk cell expansion, layering on their own unique IP into the front end or back end of the process, and then allows them time to focus on product development and less time and money on process development, saving them millions of dollars and years of time within the context of getting a product concept from ideation into the clinic. Rooster Bio chose MSCs as our proof of concept because of their wide breadth and large clinical focus, their multi-lineage potential, immunomodulatory features, ability to be biofabricated into engineered tissues and organs, or even genetically engineered, make them an interesting and powerful cell type with an established clinical history. With the number of late-stage clinical trials increasing rapidly, it's a matter of time before MSCs have their moment. The built-in supply chain and processes created by Rooster Bio should enable researchers to fully harness this power at a faster rate. To enable MSCs to reach full potential and to fuel regenerative medicine therapies, we have established off-the-shelf, high-volume working cell banks and highly efficient paired medias that come with bioprocess recommendations to rapidly enable customers to get into scalable flask-based or bioreactor-based production processes for bulk MSC expansion or EV collection. We have 3 grades of product: serum-containing research grade, xeno-free development grade, and cGMP clinic control grade. Our integrated product systems all work as standalone products, but their greatest power is when used together as a system. That's when we can generate billions of high-quality MSCs or trillions of EVs. That said, an autologous therapy would likely benefit from our bioprocessing media alone, and an EV program would benefit from our EV solutions, and a GE program would benefit from our genetic engineering media. Each have the potential to provide tremendous value. On the far right of this picture, we talk about our development services. And in order to speed progress, we've built these capabilities to allow for our industry-leading knowledge and products to accelerate your development, whether that be scaling out 2D processing, transitions to 3D bioreactors, looking at EV production optimization, donor comparability studies, analytical assays, etc. These products and services are being used around the world today. And while we have many examples showing how an industrialized supply chain impacts speed to clinic, there is one particular case study worth highlighting. Here is a study from a well-known CGT company looking to adapt their proprietary cell engineering platform into an MSC therapy designed to treat COVID-induced ARDS. This company had no established history working with MSCs. And the challenge they faced was to meet the urgency for a rapid COVID response by selecting both translation-ready materials and creating translation-ready processes. Ultimately, they selected Rooster Bio processes and products and they were able to drop our high-quality plug-and-play solutions into their established development program. The outcome was within 6 months, the company had submitted an IND and received FDA approval to begin a phase 1 trial, which might be a record. The company took 3 months to design a successful candidate and another 3 months to develop a robust GMP-compliant manufacturing process. This example highlights how a clinical stage biopharma company with an established cell therapy program could take a cell that they had no prior experience with and rapidly implement a program around it. There are others with similar results. And when we look globally, we have that was just one example from the hundreds of accounts, both government, academic, translational centers, and industry partners from 25 countries across 6 continents that leverage Rooster Bio platforms to assist in their therapeutic programs. And on behalf of Rooster Bio, our growing team of passionate employees located in Frederick, Maryland is proud to fuel the rapid commercialization of scalable regenerative cures, and we are eager to lend our support. Stay tuned for more radical innovations meant to disrupt the norm and accelerate therapeutic development. Thank you.